Not getting any arm comms. It's not good. Do you know which box is there for the arm under there? Shine a light down there, I'll show you. It's under your legs, Stephen. There's a craft box there. The big gray one in the middle of the rack. So on and off switch on that. Yeah. Turn it back on. Did you turn it back on?
I did, yeah.
Okay, never mind. It's slow coming up. All right. Zoom in there a little. Okay, that's good. Sorry. Blind angle.
Yeah, it's tough. Nice.
Okay, can you do the, Kylie, throw up the slurp camera?
Sure.
It's on the flush jar, so just turn it on 30 or 40%. Start to see something moving around in there.
Okay. Flush jar, starter section, 40%.
And another 50%. See it moving there now.
Got 50. There you go.
Okay, so shut that off, shut the pump off.
Okay, pumps off.
Go around the number 2.
To sample jar forward. Oh, I always do that.
Other way, yeah.
Sorry.
Well, it's kind of
There we go.
perfect. And then let me get down there, and then at 30 or 40%, it should be good.
Sounds good.
Okay, you can turn it on now.
Got 40.
Try a bit more.
Got 50.
Might be a bit much for it.
Got 60.
All right, that's plenty. Yeah. You guys want to try and put this in a sample box?
Yeah.
Yeah.
I don't want to. I don't
You
want
wanna
to
turn
try and
off the
jam it in
suction?
there. I might
Yeah.
plug it up.
Yeah.
No worries.
Section's off.
Roger.
Do you want it in the front?
Where would you guys like it?
How floaty do you think?
It's pretty floaty.
Yeah. Why don't we put it in? Do you think we're going to end up with 8 rocks?
Hopefully, yeah. It'd be nice
So-
to save some of the bigger
Okay.
compartments for rocks.
So we could put it in with the sponge.
Okay.
Okay.
So that's forward A, port side.
Roger. Toolbox out, please.
Toolbox out.
I missed what we were thinking this was. Jamie, is this a black coral?
You know, you missed it because it never happened. I'm
Ah.
not sure. I am seeing the darker part of that stalk,
Yeah.
so black coral is a good assumption. But I'm not sure.
All right.
When you're ready, I'll tool tray in.
Get out of here. Okay.
There you go.
Nav, can you drop a target that was 038?
Will do.
Nice one, Mike. Thank you.
No sweat.
A nice assist, Kylie.
Now, can we bring the sample
Yep.
carousel around to the flush again?
Back to flush.
Roger.
Alrighty.
And the pump is off, right?
Pump is off.
Yep. Okay. Great. All right.
Good to
move
go?
ship. Yep.
Bridge nav. Request we continue tracking our line. Thank you.
And as we're moving forward, I'll just say again, thank you to everybody watching on nautiluslive.org. We are exploring a area that we are calling Dragon's Back Seamount, south of Palmyra Atoll, here in the Pacific Remote Islands Marine National Monument. This is the third dive of this leg of the expedition. Total, this expedition is about 3 weeks of us being on the ship. We spent about 4 days transiting to our first location at Kingman Reef, and we'll be, spending time diving on these deep sea features in the Pacific Remote Islands Marine National Monument.
Oh wow, just ends right. No, never mind, just a patch.
It did look like it went on forever there for a second. Even still in Herc view, it does look like that. But if you check out Argus view, you can see that patch from above. Just a reminder to our viewers that you can switch between a few different viewpoints. Get that Herc camera, which is that main camera on Hercules. Also that Argus view, so above Hercules, you can see the context of the area that we're in. And then on quad view, you also get a peek into what Chris is looking at there with the positioning of the ship and the vehicles. And the live streaming of these dives does serve a few purposes. When we're live streaming to public audiences like you, it's enabling us to share something that not everyone gets to experience, obviously, and also to answer your questions, which is really awesome to be able to add that layer of of engagement on top of it. But we also have a ton of scientists ashore who are not out on this cruise with us who watch these dives and follow along with the stream. So those are big reasons that we do this.
Can we get a zoom in on the little white patch in that rock? Is it?
Yeah, just creep in there. Come up a little bit on Argus there. Go ahead, video.
Looks like another one of those
Yeah, it's just it's bigger than some of the other ones.
things.
So.
Sort of like a fan.
Full zoom.
Roger.
It's got a nice little cave there.
Yeah. Would that be a collapsed pillow? That little area?
Probably, yeah.
Okay, wide. There's another one over here. One to the left somewhere. I thought there was. Maybe not.
Hey Jamie, I've got a question coming in about coral. I don't think I know the answer to this. Maybe we can have at it together.
Go for it.
Our friend is wondering about the growth of the corals and whether it's a single individual that clones itself, like all the little polyps, since the polyps are little individual polyps, or is it multiple individuals that build the stock together? I guess the differentiation would be the genetics of of the different polyps.
Yeah, that is a good question. Let's think about it a minute.
Yeah, we can. We're also. We'll see if we don't come up with an answer, we can talk to Steve and get that answer for you later.
There's some kind of greenish critter at the base of that boulder.
Oh yeah. Zoom in there, video.
That be some sort of hydrozoan?
It's a little worm up toward the top of the screen. Just off screen now.
Oh yeah.
It's full zoom.
It's hard for me to tow in there. A couple of big rocks in front of me.
All right, thank you for the zoom.
Hey, a couple things on this rock here.
Yeah. It's just not calibrated properly. You can ignore the stops and do it, but it's hard to tell when it's level from here. Like when it's level, it doesn't look like it's level in that camera. Yeah. Yeah, write it down on the sheet for now. We'll hand it over. Zoom in there.
Oh, sorry.
That one probably is a black coral.
You can see the dark central stalk there. And so when we refer to black corals, we're talking about corals that instead of having a calcium carbonate skeleton,
full wide for a
have
minute.
a
Hung
darker
up here.
skeleton made of protein. And it looks like there might be a brittle star wrapped around a sponge to the right.
Are you interested in sampling the coral?
Not the coral, no.
Okay.
Though there's a zoom on the stalk, yeah, would be great.
Well, Roger. Okay, video.
That's cool.
Oh,
Yeah.
it's
Pretty
a
neat.
broken off stalk or something.
Yeah, it kind of looks like something we sampled, in which case that would be a glass sponge stalk.
Mm-hmm.
Coming in a little tighter.
Yep.
Yeah, I would say that's a sponge stalk with a very clingy brittle star. Thank you for the zoom.
All right,
Coming wide.
full wide.
Jamie, I've got a sampling question for you.
One moment, I'm going to take a few notes about what we
Yeah.
just looked at.
Do what you gotta do.
All right, I am ready.
We got some folks wondering about when these samples get brought to the surface, what sort of effects the difference in pressure has on them.
You think any of those rocks are loose right above the lasers? Or they...
You want to take a poke around?
Yeah, that'd be great.
All right, stop ship here.
Bridge nav. Request hold position.
Full wide.
Thank you.
That's full.
So yeah, we'll see how much of this we can fit in between potential sampling, but the types of organisms we sample are pretty much exclusively invertebrates. We don't tend to sample fish most of the time. So these animals don't have air sacs in their bodies. Like human lungs, for example, would be an air sac. Fish swim bladders are also air sacs. So because you don't have these air pockets,
Sorry, just a second.
Yep.
Kevin, which rock are you after?
Maybe the one right where the laser's on right now.
Okay, zoom in
They
there.
kind of look in situ though, so I don't know if any of them are going to move.
Yeah.
I will resume after we finish up here.
Sounds good.
Okay. Zoom in a little. I don't think that's moving. Oh, there's that little guy moved.
Oh, the one right,
The one
below
right
you
under me
moving.
is
Yeah, might as well
good.
grab that one.
All right.
Kind of has a thick manganese crust, but I'll hope for the best.
The larger one in the upper left looks pretty angular too.
Yeah. Let's see.
Upper left, yeah. You
Yeah, we'll
wanna-
see if we can grab that one too. Oh,
Oh, that was an accident.
well, it worked out.
Sit, stay.
Right above the claws.
Oh,
bigger one.
up here.
Yeah. If it moves.
Ooh.
Oh,
Surprisingly flat.
Yeah. Dramatic foggy intro to the rock.
Keeper?
No, I think the fatter one's better. This one might be all crust. It's hard to tell.
I like that one.
Sorry, Jen.
Its flatness appealed to me.
Which one are we after?
Either the one we grabbed originally or the one right to the left of it.
Okay. You have a preference?
Let's try the one to the left since I haven't seen that one yet.
nice.
Yeah, I'm digging this one. Nice and fat.
Come in a little tighter here for this.
Is it just me, or did us? Was it a brittle star like fell off from under it
I think it did.
on the left in the fog there?
Yeah, that's keeping.
Alright, if we could put that in the starboard bio box, that would be great. I'm assuming that's big enough that it needs to go in one of the larger compartments.
I think so.
Okay, so either one of the large compartments on the starboard is available.
Okay, you show me the arm in the bubble cam.
Run the tray out now.
Yep.
How's that?
It's good.
Check out the quad stream to see what's happening on the starboard side of the...
We said either one?
Either one is free. Your choice.
Low hanging fruit.
You think that'll fit in a small one?
Yeah, you want to try it?
Okay, coming out
If
more.
it could fit in a small one, that would be great. Let's see.
is the only
We
one
have
with stuff in
something
it,
in
right?
D, the aftmost compartment, so maybe could we try
close.
for? Yeah,
You want? Is A okay?
A would be fine if it fits.
Okay,
Awesome.
Like a glove.
Thank you very
Thank
much.
you.
Okay,
kindly.
dual tray and, or sorry, sample tray and.
Nav, can you drop a target? That was 039.
039.
It's slow going.
Alright,
All set.
all done with the box.
Which box?
The starboard box.
Oh, sure.
Yeah, we're ready to move the bubble back over the porch.
Bridge nav. Request we continue on our line. Thank you.
Yeah, it's that cathode light.
It's all set now though? Oh, never mind.
Yeah.
There it is.
Yeah, it's coming in and out.
Yeah, I got it.
Okay, where were we in our story?
We had mentioned that most of the things that are collected are invertebrates.
Oh, okay, good. And then we were talking about air sacs. Okay.
Yes.
So because we're collecting invertebrates without air sacs, they don't tend to change form when we bring them up. So for example, if we brought up a squat lobster or a coral or a sponge, it would look basically the same on the bottom as it does on the surface when we take it out of the compartment. It doesn't really have visible effects from the pressure change in terms of its body structure. So there aren't air sacs to compress, and so it stays about the same. Same is true for also the very gelatinous sea cucumber we collected the other day. It's mostly water filled, so the water doesn't really compress or shrink to any great degree. So it looked the same when we brought it up as it did swimming through the water.
Awesome. Thank you, Jamie. And another note about samples, we are in a marine national monument, so we do have parameters around what and how much we can sample, but we also are constrained by what we can fit on Hercules. So we've got the bio boxes in the front, bio boxes on the starboard side, and then our slurp containers. And then the water samplers. And am I missing anything?
Nope. On
Yeah.
this particular series of dives, it's just what you mentioned. On many Nautilus dives, we also have
There's
push cores,
something on the left.
but we're not taking those for this cruise. They're not really of focal interest to our relevant scientists.
Zoom in
I'm
there,
gonna
video.
guess that this is another sponge
stock.
stock.
Yeah.
And we're seeing brittle stars at the top of these.
Full zoom.
Yeah, it
that
looks
are
like at least 2,
maybe
if not
3.
3. Yeah, maybe 3. I'm leaning toward 3.
And so they're lifted up into the water, allows them to catch more of that marine snow that they're eating. Look at those wiggly arms.
All right, all one.
Oop, it looks like another black coral to the right of the lasers, or right where the lasers are.
Oh yeah. Zoom in there.
Coming in. Full zoom.
Now, if I remember correctly from the talk that Steve gave us, he had mentioned that black corals, the tentacles and the polyp structure looks much different because instead of having those little standalone polyps with the tentacles at the top, the tentacles
Okay,
are
come on.
along the stalk, and the mouth is actually along the stalk as well.
Huh.
Yeah, that's right. So, unlike the other ones where they're circular and you have a polyp with a central mouth and a ring of tentacles,
Clear for
this
gauges?
one, I believe
Sure.
he said it had a mouth and then 3 tentacles on one side, 3 on the other. So a row of 6 with the mouth in the middle.
Big sponge on the right.
zoom in there?
Ooh,
Oh, cool.
cool.
Nice
A
one.
little store, slime store or something on the bottom.
Yeah, I see the little pink star that just went off screen.
This is full zoom.
It's pretty cool. It builds like a little web to catch things.
That's so cool. And so again, if I'm remembering correctly, those little strings that we're seeing coming off the side is helping to grab that food that's actually being funneled through the top of the, or through the, it's coming through the top of the sponge, I believe, and being forced through those side holes where the little sponge is consuming things.
Very neat one.
Oh, and a shrimp decided to come hang out too.
Very long legs.
Hey Jamie, we've got a follow-up about the pressure change on the samples once you're ready.
Okay, 1 second. Okay, ready.
We've got a viewer wondering if the lack of change at the macro scale of these organisms holds true at the molecular scale. Is there any cellular change at change of pressure?
That's a very good question. I believe there is, so the change in pressure and temperature is kind of stressful for the organism. So some scientists do research where they want to keep the animals alive and intact and take them back to their laboratories to do studies. For example, reproductive studies with deep sea mussels is one that I know that happens sometimes.
And Steve's lab at Temple University, the Cortez lab where Steve works, they do experiments on live coral that's been collected.
Awesome, yeah, so that would be another example. So I believe for organisms that are found around 1,000 meters or shallower, it's not too harsh on them to come up to the surface and be maintained alive for those types of long-term studies. But for animals below 1,000 meters, if you want to keep them alive long-term, it's better to bring them up in a pressure chamber so that they don't experience as much stress in the change in pressure and temperature when they come up to the surface waters. Let's see, so yeah, that also is relevant at the genetic level. So a stressed organism will start to make different genetic proteins and have stress responses at the cellular level. And so some people that study active gene transcription and that type of thing would want to make sure they kept the animal as unstressed as possible if they wanted to know how the organism behaved in situ and the types of
Full zoom.
functions that organism had on a normal basis. So that would be another good use of a pressure chamber, is to look at active genetic code transcription and that type of thing.
Cool. Thank you, Jamie.
No problem.
We are just about at the top of this first peak here.
Gonna probably try to grab one more sample from this top of the peak where it's nice and thick volcanic.
Yeah, a lot thicker.
Big change.
Would we call what we're looking at right now a truncated pillow?
There's a coal.
Yeah, cool.
Not
Not quite.
quite. These are more massive flows.
Got it.
Do a zoom there.
Big pillows though, for sure.
Ooh, pretty.
Just full zoom. There's another one of those leaf-like
Yeah,
things.
so this looks like the one that we sampled, and on second thought, I'm inclined to say it doesn't look like the black coral.
Yeah, it does not in this shot.
I was trying to remember if our previously sampled coral had the feathery
Okay, come
type
wide.
of polyps or the single polyps along the stalk.
I think it did look like it had more of the feathery
Yeah,
type
I think
polyps.
so too. So I'm not sure if it was a black coral or not.
Here's a picture in the guide with those associates on it.
Yeah.
That's not what we're looking at now, but that might have been what we sampled.
Oh yeah, awesome.
I
So
think
we have a picture in our coral guide that Steve made
That's a big
us of a very similar looking, I don't know about the coral, but similar to the black corals we saw at least. We're still not sure about the coral we just looked at or sampled, but it also has the barnacle associates on it, and that's from Paulo Seamount, which
There's a big stocked one.
somewhere in this region as well.
I was just going to ask if those were barnacles.
Zoom in a little bit. It's good there. Just go past this and get the next thing.
Fancy. Looks like a stalked sponge.
Mm-hmm.
Is that full zoom?
Not quite. Coming in. Full zoom.
Thank you, that's awesome. We're
Cool.
good here.
Alright, come wide.
Oh, hello. And Jamie, I don't know if you saw it, but it looked like there was a coral that just went off screen as well near that sponge.
I did not see it. What did it look like?
It looked like it might have been Iridogorgia, but I don't know if that's just because that's my favorite one to see and say.
Yeah, I missed it, sorry. Can we get a zoom on this stock?
Yeah, go ahead, video.
Another brittle star apartment.
Full zoom.
All right, that's great. Thank you.
Michael, do we have on more light on Hercules than we usually do?
No, just a regular show is on.
Can we get a zoom on that coral, I think?
Yeah, go ahead.
Coming in.
A twofer.
Nice.
Well, that looks like a black coral again.
Yes, black coral with a pretty active unstocked crinoid.
That'd be really cool if it would fly for us.
Oh, it's
Release.
maybe.
His little feet look like they're still holding on pretty good.
Pretty tight. Just stretching.
Oh!
Decorator crab.
Yeah, hermit crab with an
Coming wide.
anemone on its back. It is booking it.
Go in on that.
Coming in.
Let us see you. Oh, you look beautiful.
Got a sea urchin for a butt. What? Is that normal? Is that how he looks all the time?
Anemone. They put things like anemones on their backs as camouflage and also protection because the anemones sting.
Anemones.
That is cool.
Callback.
So he's maybe 5 centimeters based on the lasers. Maybe a little bigger.
I love the way these look.
He or she.
I don't know what you guys are talking about. I can't see it.
Perfectly camouflaged.
I just see an anemone.
That was cool.
Undersea camo jokes.
Still seeing more of those little, like, protrusions from the seafloor.
Looks like a sea cucumber.
Different
Yeah, it
looking
looks
one.
like it.
Can we get a zoom on it?
Yeah, go ahead.
Can see those tube feet at the front that are taking food in. And you can also see all of that sediment in its digestive tract.
Oh, here's a better look.
Oh.
Again.
I don't like.
You didn't like that? Wow.
Well, this one's got like that little like,
Polychaete on its butt.
well, the like little frilly hood thing.
Oh, yeah.
Well, at least he's eating good.
That was amazing.
How majestic is nature.
Can we get the lasers off for a minute?
Truly.
Yeah.
Wow. It's a really nice shot. And so it expelled the sand to sort of propel itself up from the seafloor. Now it's just kind of wiggling to swim. It's pretty cool.
Nice shot. Thank
Yeah.
you.
Bye, buddy.
Very cool.
That was sweet.
You can toss the lasers back on if you'd like.
Sure.
We're just about at the top here.
So for those viewing at home, the sort of squiggle that you were seeing inside of the sea cucumber is that it was translucent, and we were seeing into its digestive tract there.
Chris, can you zoom out on high pack so waypoint 3 is in view, please?
Yes. Waypoint 3 is actually named Waypoint 2 as well.
Oh, that's clever.
But because that's the way numbers go. But yes, I can.
Is there a way to adjust that?
Yeah, I can.
Okay.
I can change that. I just want to make sure that the next one isn't Waypoint 3.
The next one's waypoint 4.
Next one's waypoint 4?
I'm pretty confident.
Okay, I can change that then.
I was looking at the HIPEC plan, and
Yep.
that one before didn't have a 3, it just had a 4.
All right, I will adjust that because I can count.
Yes, you can.
1, a 1, a 1, 2, 2, 4.
We got Steve here for video engineering, Folly sound effects, and a soundtrack.
Yeah, there was no fluttering with that sea cucumber.
Oh yeah, where was the Folly sound at that one, Steve?
I failed you. I thought it was best not to follow the pooping.
That was probably a good call.
But if you want.
All set.
I think we're good.
The moment has passed.
Yeah, but I think that might have taken away from the majesty of the moment a little bit.
Yeah, the sediment was also passed.
I hear that sea cucumbers are bad liars. You can see right through them.
Waiting for it. I was waiting for it.
Yeah.
You proud of yourself?
It's that time. It's that time of the night.
Morning?
It's the morning.
Yeah, it's light out now.
So early.
Is it?
If it's light out, but we didn't see it happen, is it really light out?
Yes.
Thank you, Allison, for keeping us
Coffee
in check.
beans.
On the rails.
Thank you.
Do we know if these sea cucumbers have many predators down here?
I don't think they do. I would imagine if something was going to eat them, it would be a large fish.
Yeah.
We have not been seeing many fish in this particular dive, though.
And then so is this sort of flat sedimented area, this is the top or the peak of the first peak, or are we past that?
Yes, no, we're just about right on top right now.
Got it.
So we'll be heading down slope for a little bit before we go back up to a larger one.
Great.
There are no rocks to sample up here.
Nope.
Nope, surprisingly smooth and sedimented.
Maybe if we go over the edge back down, change.
Yeah, hopefully we can find that massive flow again on the other side. I'll try one too. No, sorry. Looks like there's a beautiful sunrise out there.
Again, if you check out that quad view, Steve has thrown up the sunrise for us.
Yeah. Where did they go?
So while we're cruising along this sedimented area, I'll just remind you where we all are. We are south of Palmyra Atoll, here in the Pacific Remote Islands Marine National Monument. We've just made our way up to the first peak of what we're calling the Dragon's Back Seamount, because it has multiple peaks that we're going to be exploring on this dive. And thank you for joining us. And again, you can send in questions into that little box below the live feed. And we'll do our best to answer them between our operations and scientific conversations.
Are we going to want to go on the top of this little ridge or saddle here in between the two, or along the side of it? What?
Is today your takeover, Chris?
Today is my takeover. How can you tell?
Oh, no reason. Hey. What we're referring to is that on Nautilus Live Instagram, throughout this cruise, different people from the Corps of Exploration.
Can you, go ahead.
Yeah. So, do you guys want to go on top of the...
Yeah.
Okay.
If we want, we can go down the side of this slightly just to see if I can grab another rock and then jump into that. I don't know if we need to go down the whole slope.
Okay. Yeah, as we come over and start transitioning, I can change from aligned to steps.
Could we zoom on
yeah.
the
Just one for this.
left side of the screen, that tan blob?
Yeah. Zoom in there, please.
Looks like a old sponge, probably dead. And then could we also zoom, if we go full wide again?
This one?
Yeah, right near there, slightly more over. Would be great.
Go ahead.
Look like a black coral that we just zoomed past, and then another of these white branching corals that we have yet to ID, plus 2 crinoids. All right, we're good here, thank you.
Beautiful. This whitish translucent coral looks so delicate and feathery. Yeah, to our viewers
We're
at
going
home.
down slope now, so we can just swing around 180, and it'll be easier to go down, back down the hill. You can go to either side. Yep.
But if you're interested in getting a behind-the-scenes look at what life is like on the ship, make sure you check out Nautilus Live on Instagram. And many people from the Corps of Exploration will be doing Instagram takeovers to give you their perspective. And today is Chris, our navigator.
Yep. So I will try my best to highlight the different jobs on board. That we have 2 navigators on board, and myself as a navigation intern. So a little view into what we do.
Excited to see what you post. And if you, I think you should still be able to see the story from yesterday where our very own Kylie
Should be right
Pasternak
under you.
did a
Okay.
takeover.
As we drive away.
Okay, my camera's all the way down.
Yeah.
Yeah, so I did like a ROV walkthrough yesterday. It's probably still up. It was only like 12 hours ago.
It is.
Thank you. Verification. So like if you can find all the emojis I tagged like different instruments on the vehicles with, so you could see, try to figure out like where the cameras are, where the lights are, get up close and personal, because those are the things that we check for pre-dive and things like that. So if you want to be a ROV intern or ROV pilot someday, these are things you're going to be doing.
Yeah, and it just gives you even more perspective on the intricacy of like Hercules as a vehicle. It's got a lot going on.
I
And
didn't
Argus
actually
as well.
get too into Hercules.
Oh yeah.